I'm very excited to welcome my colleague here at the University of Washington in the Department of Psychology, Jen Forsythe. If you think neuroimaging is complicated, genetics is complicated, putting the two together is really, really complicated, and which is what Jen does, and also will tell us a little bit about today. So thanks, Jen.
Yeah, thanks so much for the invitation and for having me. I hope you all have been having a fun week so far, learning plenty, but hopefully not too much that you're not absorbing. So yeah, thanks for having me. So just before I do anything else, I just want to do quick acknowledgement. So I'm not going to present a ton of data from my own lab today, but I want to give a shout out to my lab here, the Genetic Risk and Neurodevelopment Lab, where we're working on a lot of questions related to this currently. And then when I do show a little bit of our own data, it's actually going to be from some work that I did when I was a postdoc a few years back at UCLA. And so I'm going to be showing some work that I did as part of the 22Q 11.2 Enigma consortia, where we were working on an imaging transcriptomic project. Okay. So just a little bit about me and my own research interests. So I am a clinical psychologist. So that was what my PhD was in, but I've always been really interested in disease mechanisms. And in particular, I'm really interested in serious mental illness and how it develops and what are the mechanisms that underlie that. So for me, I've always been kind of agnostic to whatever methods are going to help us get a grasp on that. And so for that reason, that's where for me, neuroimaging has been really useful, and there are of course limitations to it, like with any other method. And genetics has also been really of interest, and is there's a whole lot that you can do with genetics, and it's a really powerful tool. So I've increasingly incorporated genetics into the work that I do. And my main objectives for today with all of you is to make the case for why it's worthwhile to think about genetics and related transcriptomic data and linking that kind of data to neuroimaging data. So what does that give us? I also want to give you just some background. I'm assuming that most of the folks here have a little more background in neuroimaging, neuroscience, psychology, maybe a little less on the genetic side. So, and I know that it's going to vary for different people in the audience, but I'm hoping to get us all kind of up to speed and to make it maybe a little bit easier if you haven't been reading a lot of the genetics literature, to sort of be able to feel like you can read that literature and understand it, and also think about possibly applying some of these methods in your work. And so towards thinking about how you could apply some of these kinds of methods in your work, I'm also going to talk through some common study designs that incorporate genetics and neuroimaging, and we'll do a little bit of a hands-on tutorial using imaging transcriptomics in particular. So, okay, so what does that mean we're actually going to do? I'm going to spend the first part of the talk doing just an intro to the central dogma of molecular biology and the human genome. Then we're going to talk through 3 different research designs related to imaging genetics. So first, studies that try to find specific genetic variants that are associated with different neuroimaging phenotypes. The second one is studies that use cumulative genetic scores and try to link them to imaging or other brain-related phenotypes. And then the third one is imaging transcriptomics, where you're not using genetic data exactly, but you're using sort of the next level up. So gene expression data, that's the output of how much each of our genes gets expressed, and relating that to neuroimaging data. Okay, so why should we think about incorporating genetics or related kinds of omics data? These are some figures from the Enigma working groups for schizophrenia and depression that came out several years ago, where they were just mapping across the brain the effect size across regions in terms of patients' cortical thickness, that's for schizophrenia on the left, or depression for cortical thickness on the right. So I would love to hear from some of you seeing these results. These were super robust findings, right? These studies were done in thousands of patients and controls to create these really robust, reproducible maps of effect size. So what do you feel like you know after seeing that? Someone holler at anything. Did we learn anything from this study? Does it matter to you that, for example, in schizophrenia patients, they're seeing where like the red colors are greater group differences, they're seeing maybe greater group differences in regions of the temporal lobe, as well as some frontal regions, and maybe less regions, for example, in occipital cortex, regions related to primary sensory processing. You feel like that tells you anything? I will say that within the literature, people have, when they see that kind of literature, they're saying, okay, it seems like even though you actually see some differences in cortical thickness across the brain, that's also implicating in schizophrenia that maybe regions associated with higher cognitive processes tend to show greater differences in cortical thickness compared to regions involved, for example, in lower level sensory processing. Some of those regions that show larger changes also happen to be regions of the brain that mature a little bit later. So there's some implications there for the field when we see things like that versus, for example, on the right for depression, the overall effect size differences in cortical thickness in this Enigma study, they're much lower overall than the differences that you see in schizophrenia. And the specific regions that are coming out more strongly look a little bit different, right? So you're seeing parts of the anterior and posterior cingulate cortex, also some orbitofrontal cortex, so implicating maybe slightly different circuits in depression. Okay, so hopefully that seems like that was a worthwhile set of studies to do. A lot of effort by a lot of people to create those data sets. Now, even if we know that, what do you feel like we don't know? What does this not tell us potentially about mechanisms? And maybe we can do this as a think, pair, share, because I feel like maybe it's hard to speak up in this room. So if people want to just turn to someone around you, take maybe just a minute or two and just brainstorm, even though they found these really robust findings that implicate maybe slightly different brain circuits, what do you feel like we still don't know afterwards about the mechanisms underlying these disorders? And then we'll come back. So we'll give it just a minute, just turn to a neighbor near you.
Yeah. So
Okay, let me see if I can see. Do I have to exit out of my slides to see something? What's that? Okay, awesome. Thank you. Okay, why don't we wrap up? Okay, I would love to hear from you all, and I know there's some folks online who also maybe have some ideas. Let's hear maybe a thought or two from folks in the room.
is it immediate future environment?
Yeah, awesome. I love that. Let me just repeat that for folks who maybe didn't hear that even though we're seeing these patterns that we don't know whether it's genetically mediated, environmentally mediated, is there some kind of interaction? We don't actually know sort of what's the causal pathway to get to this spot. Love that. One more, any other thoughts from maybe one more person in the room? Yeah.
Differences.
Awesome. Yeah, love that. Right. So we also don't know, like, is this a static difference? Maybe do patients at the beginning of when they start to show symptoms have this pattern, or even before they start to show full-blown symptoms? Because, for example, the typical age of onset for schizophrenia is late adolescence, early adulthood. For depression, it's maybe adolescence into adulthood. Do you see it just with symptoms? Do you see it earlier in development? Love that. Any other thoughts that people had? Yeah. Yes. Yeah, awesome. Love that. So is it related to clinical variability and severity? Yeah, these are all awesome points. Okay. And oh, yeah, one more, go for it. Yeah, awesome. So also, like, is this, what does it look like when people have more than one disorder? Or there may be different patterns depending on sort of the comorbidity with other disorders. Maybe just do some of these, what does it look like if somebody also has comorbid substance use, for example, or maybe previously had a diagnosis of ADHD when they were a child? Great points. Thank you, everyone. And so I will say genetics cannot answer exactly all of those, but it absolutely can help us get more of a handle on a number of them. So this is just sort of on the right of the screen. It's just showing sort of a flow, right? As we think about when we're doing neuroimaging, we're sort of near to the top part, right? Like, especially if we're using like MRI, that is, non-invasive, somebody's just in your scanner, you're looking at cortical thickness, you're looking at functional connectivity. You're at that sort of systems level, sort of macro level of differences in the brain maybe associated with a trait. But if we're thinking about etiological pathways that incorporate genetics, and we know, for example, that a lot of these disorders are highly heritable. So genetics is playing a big part, although also so we're actually starting down here, right? So there's all of us carry different genetic variants. Some of them directly impact the structure of a protein, if they might be within the protein coding regions of the genome. Some of them might lie outside of a protein coding region of the genome, where instead they're going to influence how much a gene gets expressed. And ultimately, that's going to impact the transcript level, which then is going to change how much genes are expressed at the protein level. And that is where you start to have differences in the synaptic or cellular morphology or function that sort of underlies some of our macro level neuroimaging metrics that we look at that we think are contributing also to behavior. So if we want to start to try to link these a little bit better to start to understand, if we see those kinds of findings, are genetics actually playing a causal role, or are we just seeing differences that relate to some kind of confound like medication usage, or something that is associated just with disability that maybe somebody, for example, with psychosis might have had, maybe they've withdrawn for many years. And so part of what we're seeing is brain changes that reflect that. Genetics helps give us an anchor at the bottom. So for that reason, genetics can be helpful because differences in genetics absolutely can be causal and often contribute to neurobehavioral and neuroimaging phenotypic differences. They also can be used in various ways to give us a window into biology that's more precise than just the biology that we can get at the gross macro scale neuroimaging level. And what's really nice about genetics and omics data sets is that they are there's a ton of resources now for genetics and bioinformatics that we can leverage. Many of them are publicly available. And molecular genetic methods, so indexing genetic variants that somebody carries, are increasingly affordable. So like a genotyping chip, which is very commonly used now for a lot of the kinds of studies that we'll talk about here in a second. Those they cost maybe about $50 per participant, for example, and that will get you millions of sites across the genome that commonly vary from one person to the next, so it's quite accessible cost-wise. Or if you want to get at more sites across the genome, you might do exome sequencing or whole genome sequencing, where exome sequencing is maybe $300 a person, less than a scan, right? Whole genome sequencing, was $1,000, but it is rapidly coming down. Actually, depending on some of the new methods, you could get it down to probably about the cost of exome sequencing with some of the new emerging methods. So really can be quite accessible to actually directly index genetic variation in participants that you might be studying. Okay, so if we then want to think about, okay, can we use some kinds of genetics or maybe transcriptomic methods to help us get a little bit more of a handle on some of the questions we couldn't answer just seeing maybe group differences in neuroimaging? That means we also have to feel like we feel a little bit comfortable with genetics, right? So let's, some of this might be just like super review for a lot of you or not. And so to get us all on the same page, as a reminder, right, our DNA, consists of only 4 different nucleotides: adenine, cytosine, guanine, thymine. Those 4 nucleotide bases, we together make up the instructions to make a whole human being from one single cell originally, right? Our genetic code, our DNA, is carried in the nucleus of every single cell of our body. And so that's what you're sort of seeing there, right? This nucleus here, all of our DNA and every single cell in our body has that. Okay. And that DNA sequence that we carry in each of our cells, the functional unit that we think about is genes, right, which encode those proteins, which are the building blocks of all the cells of our body. And the central dogma of molecular biology describes the flow of genetic information going from double-stranded DNA in our cell nucleus to messenger RNA. So chunks of that DNA sequence is going to get transcribed.
While the iPhone usually stops at
Oh, sorry, what was the question? Okay. Okay, so, right, so we're going from that double-stranded DNA in our cell nucleus. Chunks of it, if it's a protein coding region of the genome, will get transcribed into messenger RNA, which is a single strand of RNA. That's what you can see, that little messenger RNA little transcript. And then that messenger RNA transcript, which is nucleotide bases, right, still just 4 letters, is going to get translated into the language of proteins, which is amino acids. And every, within a protein coding region of the genome, we have triplet codons. So basically 3 nucleotides in a row will code for a different amino acid. And those amino acids together create these polypeptide chains that, because of the interactions of those amino acids, they're going to fold up in very specific ways to give you the structure, unique structure of different proteins. And that's how we build all of the cells and tissues in our body. Okay, and so just as one example, this is some DNA sequence that we each carry, where once you get down here, this portion of our DNA sequence is now starting to code for the GRIA1 gene, which encodes key subunit of the ampaglutamate receptor. So that's what it looks like. Here, that together give you the structure up there, that amino acid for that AMPA receptor subunit.
Sorry, Jennifer, do you mind if I just quickly interrupt?
Yes.
I think that when you're facing out, we cannot really hear you so well online.
Okay.
I don't know if that's just me, but for me, it's breaking off when you turn around. I'm sorry about that.
No, that's okay. Okay, I will try to stay put. I'm sorry. I'll try to stop walking around so much. Thanks for the heads up. Okay.
Thank you.
So yeah, for sure. Okay, so again, so if each cell nucleus that we have carries this sequence and the sequence for many more genes, as well as what I've hinted at is that a lot of the sequence isn't directly encoding a gene. All of those sections of the sections of our DNA sequence that encode proteins, though, right? When we think about, for example, cellular and synaptic structure, this is what they're encoding, are each of those protein building blocks. So if we think about a synapse, and I know this figure is a little bit overwhelming, but I just wanted to sort of to make it really concrete, right? That those genes are encoding, for example, the scaffold proteins on the presynaptic side of our synapse that is like holding synaptic vesicles by the presynaptic side so that they're ready to release neurotransmitter when there's an action potential that comes down. And I just put, when I've given you an example, I'm like naming the genes, some of the genes that are encoding these specific proteins, or the pre and postsynaptic sides of our synapses are held together by cell adhesion molecules. Neuroligin and neurolectin are really critical cell adhesion molecules that, again, are encoded by different genes. Same thing, the structure of the postsynaptic side of the synapse. So what that means is that if there's variation in our DNA sequence that is changing whether or not that amino acid chain is going to get built the way it's supposed to, that can have a major impact on, the normal functioning of a given cell and synapse. So that's where this can be helpful to think about linking these kinds of more micro levels of the brain. Okay. Now, I'd love for people to just take a second to think about this. Does anybody have any thoughts about how long, how many nucleotide bases you think the human genome actually is? Maybe take a second, turn to someone around you, and then we'll come back and I'll hear you. Some guesses. Okay, let's hear some numbers. Just throw out a guess. Or maybe you can throw out the guess that your partner said. Or anyone on Zoom have a guess they want to share? What was that?
Makes so much sense.
So one more time. Did I hear 3 million in the room? What was? Go ahead. Sure, go ahead. How many nucleotides? Yeah, a count of how many nucleotides. I'm so sorry if that was not. You can also guess how long it is lengthwise, and we talk about that too. So either one, I will accept your guesses on either score. There's a hand back there, yeah. 3 million? Billion. 3 billion. Oh, somebody okay. Well, that is the correct answer. Ding, 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 actually. So thank you for sharing that. That is correct, actually. That I don't think, yeah, that's awesome that came out as the first answer, but the haploid human genome is 3 billion nucleotide pairs long, right? But actually, each of our cells carries 2 copies, right? One from mom, one from dad. That got combined as the egg and sperm originally as a fertilized egg and a zygote. So in reality, in each of our cells, the vast majority of our cells, aside from an egg or a sperm, we actually carry 6 billion nucleotide pairs, which, if it was stretched all the way out, is actually about 6 feet long, so like taller than I am. But you can also think that those 6 billion nucleotide pairs means there's lots of opportunities to have some kind of variation in there, right? Because that sequence, that code, is 6 billion nucleotides long. And just some stats, so we're kind of on the same page about the human genome. Those 6 billion nucleotides, well, really the 3 billion, the haploid one, for each one copy, it codes for approximately 20,000 protein coding genes. We also have some genes that don't directly code for proteins, but are functional in that they're genes that help regulate the likelihood that other genes get expressed. So those are non-protein coding genes, which current estimates are maybe around 8,000, but that number is sort of changing around a little more. Out of the 3 billion nucleotides in one copy of our genome, only about 1 to 1.5% of that sequence is directly encoding those proteins. So there's a lot of other DNA sequence that we carry that does not directly encode those proteins. Some of it, like I was sort of saying, some sequence is relevant for changing how likely it is that a given gene is going to get transcribed into RNA. And there are other parts of the genome that we don't actually know if it has any functional purpose, really. It might just be sort of junk DNA that's sort of been around and mostly gets silenced and just doesn't get expressed into very much. But that's also an area of research trying to understand some of these large swaths of DNA that don't directly, that we haven't directly figured out what exactly it's doing. And from one person to the next, we share about 99.7% of our DNA sequence. So on average, each of us is extremely similar. At the same time, if we've got 6 billion nucleotides across our 2 copies of the genome, like I was saying, that can still give you a lot of different variants that each of us carries. Most of the variants that we carry are ones that are commonly found in the population. So ones that many other people in this room will have. But each of us also carries variants that are rare. So maybe ones that are even unique to us, or even because we have new mutations that get introduced in every egg and sperm. So that means that each of us also carries de novo mutations, for example, that neither of our parents even had. And so again, while most of the variants in our DNA sequence are ones that other people in the population will have, we do have rare variants. And on average, rare variants in the population tend to have a higher impact because they're either rare, because they're, like I was saying, newly arisen, so they haven't been subjected to selective pressure through generations of reproduction, or they're actually under negative selection. So they're being selected against with reproduction and subsequent generations. But again, even with all of those variants that we do carry, not all of them are actually going to have an impact on the phenotype. Whether or not they do depends on where they're located in the genome. So like I've been hinting at, are they actually located in a region of the genome that encodes a protein, or are they located in a region of the genome that maybe is regulating the likelihood that a protein is going to get expressed? And if not, it might have very little impact on a phenotype. So just to drive this home a little bit more, what I've been hinting at is that in terms of gene expression, each of our cells has pretty different profiles of gene expression. So for example, from one tissue or organ compared to the next, the amount of variation in gene expression that we see, like about 47, something like that, 40 to 50% of the variance in gene expression across our cells will be from like one tissue to the next, compared to maybe about a few percent is like the differences we would see from one person to the next. And it is the selective expression of different combinations of genes that determines the function and the characteristics of these different cells. It also is part of what drives pluripotent stem cells, so during fetal development, for example, to turn into some of our different cell types. So for example, to turn into an oligodendrocyte on the left versus a neuron. In the middle, you're going to see different levels of genes get expressed in different ways. And those differing combinations of gene expression per cell is also what gives cells their stable identity. And part of why this matters that I'm hinting at is also when we get to imaging transcriptomics, I think it's important to understand this because this gives you context for the kinds of findings that people are seeing. Okay. A little bit more just about the kinds of variants that each of us carry. So this is a schematic from like a really classic paper from like a decade and a half ago now, showing the average amount of variation you'd see from one person to the next across 5,000 bases. So each of these spots, each of the columns is showing you a spot where there's some variation among these 10 individuals. And in between, there's other nucleotides that aren't different between them. And again, because we each have 2 copies of the genome, that's why there's 2 rows per person. Most commonly, what gets analyzed, because it's been pretty cheap and the methods got developed pretty quickly, are common single nucleotide polymorphisms, which is that second row where people either have a C there or a G there. So do you have a cytosine or a guanine in that spot, in that copy in each of your copies of the genome? So those are SNPs. Which you've probably heard people talk about. But as you can see from this plot, all of us actually carry other classes of genetic variation as well. They just haven't been analyzed as much because the methods maybe took a little bit longer to develop. Also, if they're rare, then you need larger sample sizes potentially to index them. Although people absolutely do study these other classes of variants. So I'm just going to highlight, for example, at the bottom, you can see person 10, one copy of their genome, they've actually got a chunk of DNA that is pretty big where there's just nothing there. So that's like a rare copy number deletion, for example. So that person, if that is a section of the genome, for example, that encodes some genes, that means that person's only going to have one copy of the DNA sequence for those genes. And so those genes would probably be expressed about half as much as what you might see in a typical person. Okay. So when we think about incorporating genetics to imaging, what are some of the different study designs that we commonly see? So hopefully some of you have heard of this phrase, genome-wide association studies, or a GWAS study. This is a really brute force method that now has been around for about a decade and a half, and it's been wildly successful in many ways. I caveat to that, not that that tells us everything, but it's been very powerful and has given us, in terms of finding associations for complex human traits, and also allowing us to do really interesting analyses to get a window onto biology. So in this brute force method, what the researchers are doing is, if you have a genotyping chip where you are assessing common SNPs. So these chips are created to basically index spots in the genome that commonly vary across individuals. They're often testing millions of them genome-wide to basically say, is the count of whatever allele somebody has at each of those sets of millions of locations, for example, do individuals that have depression, so cases maybe in blue have depression versus controls in green. Do cases with depression, what count of Cs do they have at a given spot compared to the controls? And you're basically just counting that up. So does somebody have 2 Cs at one specific location? Do they have 1 C and 1 G, or do they have 2 Gs? And so it's just, it's a linear regression or a logistic regression millions of times over. That's what's happening in GWAS studies. And again, they're generally testing an additive model. So just saying, you know, assuming that the count of a specific allele that you have, assuming it's linear, and just, you know, do you expect somebody who has 2 Cs to be twice as likely, for example, to have depression compared to somebody that has 2 Gs at that location? And the power of the GWAS, so how successful it is in finding these associations, depends on both how much variance in a given trait is due to common variance, as well as the overall sample size, so how well powered they are. Is there a question on Zoom?
Yeah, sorry, I just wanted to ask a quick question, if you don't mind.
Yeah.
So when we do GWAS, of course, these are also cross-sectional studies. So we are looking at concurrent associations. So I guess my question there would be, even though these are genetic associations, there is still no direct way to assess causality if we're actually taking concurrent evidence, correct?
Yes, that is a great point. So this is a great comment that it is just testing for association. So this can help you, and this has been pretty powerful for like giving us associations that then, as people follow up using bioinformatics tools to sort of understand, you know, is there a convergent signal onto specific genes or biological pathways across genes? At the same time, to truly know, you'd have to do a follow-up experimental study, which people sometimes do, you know, either in animal models or, you know, maybe like induced pluripotent stem cells to try to follow up to directly show, okay, now if we sort of change this particular variant in a model system, what is the phenotype on the brain? So technically, you, to really 100% show causality, you would have to do that. Yeah, it's a good point. Okay. Nevertheless, even though there are some caveats, that's a great point that this is still correlation with the trait, right? But the nice thing about correlation with the trait at the genetic level is it's sort of one directional, right? If you see some kind of association with genetic variants, it's much less likely that you're tapping into something that's associated with downstream consequences of the trait, right? Compared to if you're looking at imaging where it's less clear, are you just seeing differences because maybe of the medication that that group of patients is on? So it's still, it helps us. Okay, so these genome-wide association studies, what are the questions that they're asking? Essentially, it's just genome-wide, what variants are associated with variation in a trait, so a brain imaging trait, the likelihood of seeing a disorder. And then again, with follow-up analyses, people look for convergent signal to understand what's the biology that's implicated by associated variants. What this actually outputs, the very basic output is these associations where you get effect sizes, so either a beta or an odds ratio for millions of spots in the genome, and the p-value statistics. So this is just like a Manhattan plot, which maybe some of you have seen, right, where what you've got, each of these dots is a different SNP that was tested for association. In this case, it's with schizophrenia from a really classic study from 2014. The genome-wide association line is the red line. So basically all the SNPs that are above that red line showed significant association in terms of a p-value from the linear regression or logistic regression. And then what often also, sorry, I'm gonna stop walking away. What you also get on the right, oftentimes the groups that do these GWAS, and these are sort of publicly available data sets that they then often make publicly available, are the summary stats. So you can go and look at all of those millions of spots in the genome, what was the effect size? And we'll get to it in a second, but that becomes helpful if, rather than doing a GWAS, you might be interested in like creating a polygenic risk score for a given trait or a genome-wide polygenic score for a trait. Does that make sense? Okay. So just as an example from GWAS studies, especially as in very recent years, some really large scale data sets that had both genotyping data and brain imaging have become available. I think both of these studies are from UK Biobank, which I'm not sure how much you all have talked about, but you know that was a study of about 500,000 individuals, a really large portion of whom had neuroimaging as well as genotyping and actually sequencing. On the left, this was a study that came out a few years ago where they did a GWAS of cortical structure in terms of surface area and thickness. And by doing this, they were able to identify so not just the associations, but then they did those follow-up analyses to look for convergent signal onto different genes and then biological pathways and identify, for example, that differences in sort of the surface area in the brain seems to be influenced by cell cycle parameters during fetal brain development. Whereas on the right, this was a paper, a GWAS that came out maybe a year later, also using mostly UK Biobank data that did a GWAS on major diffusion metrics. And then, you know, again, with the associations that they saw genome-wide, looked for convergent signal. And, you know, some of what they saw is kind of what you would expect. Like some of the genes that get implicated in white microstructure implicated like oligodendrocyte cells, for example. This has been helpful for helping us sometimes recapitulate things we already sort of suspected, sometimes also get leads on biology that maybe we didn't expect for what might be underlying or different brain traits, or also brain-related traits where we know that there are differences in the brain in different groups. So like in schizophrenia, or maybe neuroticism as a dimensional trait, or Alzheimer's disease. GWAS have also been done for a lot of these kinds of traits, which means you also have those summary statistics that get generated and made available to researchers most of the time. Okay, so just to highlight a big theme that has come out of these GWAS studies is that you do have to look genome-wide and do a systematic sort of screen to avoid spurious findings, and you have to do really rigorous multiple testing correction. With complex brain-related traits, generally, we're finding that they're highly polygenic, involving hundreds to thousands of genes. The majority of common SNPs, right, that's what they're analyzing, most have small effect sizes. And some of you might remember the candidate gene era, maybe not. That was sort of like in the 2000s, a really like popular strategy was to like say, you know, we think this gene or this small set of genes is associated with a trait, and then just to look for whether or not variance in that maybe 5 set of genes is associated. Once we started looking genome-wide systematically, we discovered that the vast majority of those candidate gene associations did not pan out. So those were spurious findings. So they largely discredited those kinds of candidate gene studies. And sort of as I've been hinting at, in order to do a GWAS though of a brain trait, oh, go ahead. Yeah, I mean, I think it's kind of the same issues that come up with any study where it's a smaller study, right? Where like, what is it, like winner's curse, p-hacking, like just things that sort of like, you know, when you're only looking at a targeted set of SNPs, you're just maybe more in a small study, you're more likely to see a stronger association. But when you actually look genome-wide, those aren't the largest signals, or oftentimes you wouldn't see an association at all. Yeah. Yeah.
While you're answering questions, there was a question on Slack, so I thought I'd ask you.
Yeah.
Nancy asks, do cases and controls need to be somewhat matched by genetic ancestry to make comparisons? Perhaps a variant acts one in a group of common ancestry versus another group?
Yes, great question. And I'm not getting into the ancestry stuff today, just because that's like a whole nother piece of it, but absolutely, like along with researchers realizing that they had to look genome-wide, systematically to have robust findings, and that they needed generally for complex traits that like a lot of us would be interested in, they really needed really large samples, like tens of thousands to hundreds of thousands of individuals. You also have to be very careful about ancestry differences if you're looking at cases versus controls, for example, because there are like very small differences in the allele frequencies, as you know, you have genetic drift, for example, like a population that has evolved maybe on an island that has been isolated. There's going to be slightly different allele frequencies in that population than maybe a population in France, for example. And so if all your cases came from that small island versus all of your controls came from France, you're going to see allele differences in the allele frequency that have nothing to do with the actual traits. Yes, you have to be super careful about matching. And the field is generally very careful about matching. Yeah.
Thought a little bit one thing that I've been struggling with is like heritability estimates.
Yeah. Yeah.
That seem to vary. Like, so I'm looking at GWAS studies and oh, this explains like 2% of variance like that.
Yeah.
And then in a twin study on the same trait, we say, oh, it's actually 40% heritable or something. And so
right.
I how do you like understand that?
Yeah. Yeah, so that's a great question. So heritability from a GWAS study is basically asking what's the amount of variation in the trait for the people who were in the study, that variance in the set of SNPs that we looked at, these common variants are explaining. So that's one very specific analysis that's restricted to just the common SNPs that they tested. Twin studies have a totally different design, right? Where it's like looking for the correlation between monozygotic twins versus dizygotic twins and saying, okay, if you see a greater correlation in phenotype in individuals that have the exact same DNA compared to individuals that share half, there's a formula to basically compute how much of the variance seems to be probably about genetics as opposed to maybe environment and other sources of like error. And that twin studies get you to like the top estimates of heritability, which would include not just common SNPs, but lots of other classes of variants that each of us carries. And, you know, there's some critiques of twin studies with those heritability estimates, right? Like, are the assumptions that the amount of environment that's shared between monozygotic twins is basically equal to dizygotic twins? You know, that's an assumption in the model. That's probably not true. That's part of the discrepancy, but I think for the field, a lot of what they also think is that the gap between what you see from SNP heritability and a GWAS versus twin heritability is also just like, we're not indexing all of the variants that people actually carry. Does that help? Okay. Okay. So to do these GWAS studies, what do people need? Again, tens of thousands to hundreds of thousands of individuals with DNA samples and phenotyping. And then also you need that genotyping array, which is indexing hundreds of thousands of spots, which then get imputed to millions, which we won't get into. But these spots, usually if they're doing a genome-wide association study with genotyping chips, which I mentioned are really quite cheap, they're going to look at just spots in the genome that commonly vary between individuals, although there's also now whole genome sequencing association studies. We don't yet have the sample sizes with that methodology so far for many of the complex traits we're interested in to find robust associations for rare variants, but people are, that is another like sort of upcoming area of work. Okay, but as you can imagine, probably most of us in this room aren't going to be the ones doing the GWAS studies, getting access to this data. Some of it is publicly available, so like through UK Biobank, but there's a lot of methods involved, and a lot of groups that are, have been actively working on that and have mined these data sets for all of these traits already. It could be that maybe some folks in the room might be interested in actually doing something a little bit different. So using some of the output from these large-scale genome-wide association studies to basically create cumulative polygenic risk scores, like I mentioned, or genome-wide polygenic scores for a new individual or like within your new study. So let's say you have a study of maybe 200 to 300 individuals and you've got neuroimaging data on them. That might be enough to do this kind of a study where you're then saying, okay, so for example, let's take all the associations for schizophrenia found from the GWAS, and now I'm going to look at what alleles each new person in my study carries at all of those same spots. I'm basically going to weight whatever allele everyone has at each of those million spots by the association of that allele with the trait to get you to one single cumulative score associated with that trait. So that's what a polygenic score is. And it started with more disease or disorder focused traits, but again, there are lots of GWAS that have been done now on continuous traits or on neuroimaging traits themselves. So you could create genome-wide polygenic scores for cortical thickness, global cortical thickness, for example, or you could do a polygenic score for Alzheimer's disease, for example. And so instead, the kinds of research questions that you could ask with these kinds of scores might be more like, is there a link between an observed neuroimaging or other related phenotype in your new study of interest and genetic variants that have been associated with that trait or maybe with another trait. So you might say, okay, in my own study, is the genetic score that somebody has for global cortical thickness, do I see that association in my new study, in this new target sample? Is it associated with any other phenotypes more broadly than just cortical thickness in this new cohort, but is it associated with life satisfaction, whether they have certain disorders? You could check things like that, which is sort of that second part of like, what other phenotypes might be associated with a given genetic score, or what is, for example, for disorders that have onset maybe later on, we were getting at this at the very beginning, if you've got a genetic risk score for schizophrenia, what is that associated with in childhood, right? Or is that typical pattern of differences that you see in schizophrenia or depression based on those Enigma structural MRI studies, are those associated with genetic risk? Or if there's not an association between somebody's genetic risk for schizophrenia or depression and that sort of classic pattern, that might hint to you that actually that classic pattern that we're seeing in terms of structural brain differences is maybe more reflecting the impact of medication, for example. Right, so you can start to do and ask those kinds of questions with these cumulative genetic risk scores. This is just a couple examples of papers that have been doing work like this with neuroimaging phenotypes. On the left, this was like a schizophrenia PRS study where the top, I would just look at A, where they're basically saying, okay, we had a cohort of like 1,000 or so schizophrenia patients and 1,000 or so controls. What's the average differences in cortical thickness that we see? That's all they did. We want to create this map so we understand what do you normally see. Then in C, what they asked is, okay, now we have a new cohort of typically developing health individuals, healthy controls who don't have schizophrenia. What is the polygenic risk score for schizophrenia associated with in terms of brain phenotypes? Does it recapitulate sort of the whole pattern of what you saw in your cases compared to your controls, or does it recapitulate parts of it? And so, for example, in their study, they saw recapitulation of parts of it, even in healthy individuals that don't have schizophrenia. So they saw differences in cortical thickness in parts of temporal cortex and frontal cortex that we do see pretty large differences among patients, but not all of the differences. Right, so it gives you some hints that maybe part of that case control difference might be pretty easy to link to sort of genetic variants associated with the disorder, but maybe some of also what we're capturing when we just see the neuroimaging phenotype is other things as well. On the right, kind of similar idea, they're looking at an Alzheimer's polygenic risk score and saying, is it associated with some of the macro scale structural differences that you see in individuals who have Alzheimer's disease? And so here they were seeing that, okay, this was again in a cohort of healthy adults who didn't have Alzheimer's disease, but an Alzheimer's PRS was associated, for example, with reductions in left hippocampal volume. Okay, and so if you want to do a study along these lines, what do you need? You need more like hundreds of individuals or maybe thousands of individuals. Ideally, if you're going to have hundreds, it's hundreds who have similar genetic ancestry. And they have to have both DNA samples and phenotyping. You need some kind of genotyping array data that has indexed those many spots across the genome. And then you just need the summary statistics from these GWAS studies of this trait to basically compute your genetic scores related to your trait of interest. And that will give you the ability to start to ask questions about whether genetic scores for different traits are associated with some neuroimaging phenotypes or related phenotypes. Any questions about that?
I have one, if you don't mind.
Sure, yeah.
I think that a question that comes up for me is, how do you decide how much of the genome you want to sample? Because surely if you sample the whole genome, that would still be pricier, especially if you want to do it with very large numbers of people.
If you're creating a cumulative genetic score of sorts?
Yeah.
Yeah, I think it's more of a, I think that like currently kind of the constraints is more like what's practical and like where do you think your time is well spent. So I think we've seen a lot of like using these kinds of cumulative genetic scores based on common SNPs. These common SNPs together for a lot of complex traits, might explain several percentages of variants in the trait, or like the schizophrenia PRS, for example, now based off the largest genome-wide association study can explain maybe like 16%, something like that, of variants in the trait. So that means then that you have a pretty well powered, if you're creating a cumulative score, a decently powered sort of single metric that you're now testing related to your imaging metrics or other metrics. And again, genotyping chips where you can create those polygenic kinds of scores are fairly cheap. And there's a lot of publicly available data also that has that. And the pipelines, because it's been around for longer, there's more documentation on pipelines and increasingly more and more tools to make it easier to compute these kinds of polygenic scores. Should you look at rare variants? Well, it also depends on which classes of rare variants. There's different pipelines for processing different kinds of rare variants where there's often maybe a little bit less documentation. If you're doing new data collection and you're trying to decide, I'm getting DNA samples and now I want to decide, do I genotype my new cohort versus do I try to get sequencing on my new cohort? Is that kind of your question?
I think my question is more along the lines of, if we know that some of these SNPs are co-localized, would we not expect that they would reflect phenotypically in similar ways? And then why would we not pick one but multiple if they're located close together?
Oh, you mean output from GWAS, how there's like linkage disequilibrium, and so like a lot of SNPs have correlated.
Yeah, or just, I mean, I'm not a geneticist myself, but even thinking about the selection to begin with, if you'd imagine that a lot of these are localized closely, then probably they're not going to encode large differences when it comes to phenotype. So then why would we not select fewer? If we can select fewer, why not select fewer?
Yeah, so most genotyping arrays that are out there to capture whatever variants people have at common SNPs, you can get pretty good coverage of the genome if you've got a good genotyping chip that maybe indexes 500, 600,000 spots in the genome because of that correlated structure between SNPs that are near one another. So you can then impute a lot of those other variants. So people will oftentimes use just, some of these low-cost genotyping chips. Like when I gave that $50 figure for a genotyping chip, that's for one of these cheaper ones where you don't have to directly index. You do like to get genome-wide coverage because there is also breaks and linkage disequilibrium, right? There's lots of variants, especially as you get further away from one another where they don't have correlated structure. So you need to index them if you want to be able to create these kinds of genome-wide polygenic scores. So I would say probably a cost-efficient decision would be one of those cheaper genotyping chips that still gives you coverage. And then the methods for creating these kinds of genetic risk scores, there's embedded methods within these tools to help you handle sort of the correlated structure. But there's also lots of variants, again, that each of us carries that are rare, where they're not so correlated with these common SNPs. So if you're interested in those variants, which again, less people will have, but when they have them on average, they tend to have a higher impact. You might have to turn to other kinds of methods, like maybe even sequencing, which is a bit more expensive to be able to capture them. Does that help?
Yeah, it does. Thank you.
Okay. Was there an, yeah, go. Yeah, that's a great question. And I would say that is a hot area of research. That's one of the questions that we're really interested in my lab. So does somebody's genetic risk as indexed by a polygenic risk score based on sort of the average association, right, in these huge genome-wide studies for schizophrenia, is that associated with severity of symptoms or age of onset, things like that? I think we're starting to see more and more studies like that, and we are seeing some signal there. We're also seeing, for example, that sometimes, again, even though these polygenic risk scores that I was talking about are mostly based on common SNPs, you can look at other classes of variants. We're also seeing that other classes of variants, if you call them and have the methods to like look at them, are also often contributing to clinical variation, but it's a really active area of research. Yeah. Okay. Okay, I'm gonna speed through this last part because I'm hoping we'll have a little bit of time for some hands-on stuff. So, okay, what if you don't actually have any genotyped subjects at all, but you still are really interested in, can we do something more with my neuroimaging data to help me get a sense of some of these more molecular cellular pathways that might contribute to it? Well, the nice thing is, that's where you can use imaging transcriptomics as one way to sort of give you some idea about more cellular molecular pathways that might be related to your trait, even if you don't have genotyping data on your current cohort. Okay, so what do you need if you're going to use imaging transcriptomics? Well, you need some kind of neuroimaging data for whatever your trait of interest is. Maybe it's individuals, case control study individuals with a given disorder versus not, or maybe it's some kind of dimensional trait of interest. And you need to know sort of the effect size of weights across some kind of parcellation of the brain. So like, how much do you see that trait is associated with changes in, for example, the thickness of different brain regions would be like one simple way to look at it. And then you can use this very nice publicly available data set called the Allen Human Brain Atlas, which is a data set that was published initially in 2012. So it's been around for just over a decade, and it's been updated since then, where it has gene expression data at really high spatial resolution from 6 individuals who were adults, who were generally healthy, non-psychiatric adults, where they indexed across thousands of different tissue samples across the brains of these individuals, how much, about 20,000 different genes, mostly protein coding genes, how much were each of them expressed across all of these different brain regions. Maybe you've seen some of the papers along these lines. They started to come out maybe around 2015, 2016. This was like an earlier paper looking at whether or not they could try to identify some of the underlying molecular pathways that might be contributing to the overall pattern of brain maturation that you see in terms of cortical thickness using these kinds of methods. Another paper that came out maybe a little bit more recently, where they were looking at, okay, what about an autism? If we have a cohort of individuals with autism compared to controls, and we look at how much, what the overall pattern of differences in cortical thickness across the brain, they could then pull in the gene expression data from parcellated to the same, parcellated in the same way to overlap with the brain, with the cortex, and then they could look for whether or not there's spatial convergence, basically. So it's sort of like a guilt by association method where you're saying, okay, if the regions that tend to show greater differences in a given cohort or associated with a given disorder tend to have higher expression of these sets of genes, what processes are those genes implicated in, or what cell types do they tend to be most important for? And that can give you some kinds of additional biological insights beyond what we were getting when we just had the neuroimaging data, right? So like in this study, for example, they found that cortical thickness differences in individuals with autism was associated with genes that are enriched for synaptic transmission pathways, and also that the same genes that show the same pattern of expression across the brain as these thickness differences are also genes that often show are found to be have lower expression in the postmortem brain samples of patients with autism. So it's a really interesting kind of finding. So what is the Allen Human Brain Atlas? I was getting at this a little bit. But again, it's a publicly available data set. You can go to that link there if you want to see about it. This is kind of just a workflow, and I won't go into the details, but basically they took these histological samples that were really densely sampled in these 6 individuals. One thing to know is that for these 6 individuals, most of them have samples taken from the left hemisphere. Not all of them have samples taken from the right hemisphere. So it just brings up some kinds of like analytical choices that people have to make when you're doing these kinds of imaging transcriptomic analyses. But the researchers at the Allen Institute, then they'd actually, before they took these tissue samples, they actually imaged these brains and put them into MNI space. So what that means is that you can then use those MNI coordinates to sort of map them onto whatever your parcellation is and find sort of the average expression in the same set of parcellations that you might be using in your own analyses to basically run these kinds of spatial convergence analyses. Really briefly, I'll just say, when the original study came out, sort of, this was just based on the first 2 individuals, describing this adult brain transcriptome, again, at this really high spatial resolution. Some of what they saw, for example, is if you've got, your brain regions, the same brain regions as rows and columns, you can see if on the upper left quadrant, we're looking at cortical samples. There's actually not a ton of when you think about all of the brain structures that they looked at, so like including subcortical structures and cerebellum, you don't actually see a ton of variation within the cortex relative to variation you might see to some other structures. But there is still some variation there. Primary visual and sensory cortex. You can see like in the middle of like the middle row, they sort of outlined primary visual cortex. You can see that it looks maybe a little more dissimilar to some of the other cortical regions. Also, some subcortical structures look maybe a little bit more distinct, like the striatum or the hippocampus maybe look a little more distinct compared to some other structures that they indexed. What they also saw that was really interesting in this paper is that if you did a dimensional reduction technique, MDS, multidimensional scaling, and then looked at sort of the top component of sort of expression, they found that the transcriptome across the cortex actually recapitulates the spatial topography of the cortex. So basically where samples within a lobe cluster together. So that's why if on the left, you've got, the different lobes as we normally think about them. And then on the right, they're just looking bottom up at the gene expression data. They're seeing that basically samples that are within a lobe tend to look more similar, be more similar in terms of their transcriptomic profiles. And not just within lobes, but also even across as you go like anterior to posterior, you see that kind of patterning, which was, I think, a really exciting finding. Okay, and that sort of set up people to start to use this transcriptional data, transcriptome-wide data from these adults to try to, use that as one way to get a little bit more information about biological processes that might underlie the patterning of differences that they see in their neuroimaging data. So I'm going to speed through this as one more concrete example kind of quickly so that hopefully we've got maybe 20 minutes to sort of do a little tutorial with Allen Human Brain Atlas. So we did a study a few years back where we were working with data from individuals who had a large copy number deletion, so known as the 22q 11.2 deletion. It's typically about 2.6 million bases long, and it deletes one copy of about 46 protein coding genes. It has a really broad phenotype that affects many tissues in the body, but it's also associated with some really important brain related phenotypes. There's increased rates of psychiatric disorders and neurodevelopmental disorders like schizophrenia, autism, intellectual disability. In terms of brain differences in terms of multimodal imaging, there's many differences, but here I'm just going to highlight some of the differences that are seen in terms of T1 structural MRI data, where on the left, you're looking at differences in surface area, and on the right, you're looking at differences in cortical thickness. And so overall, these patients with 22q deletions have really widespread reductions in surface area and actually increased thickness across the majority of regions with focal thinning in a few regions. So for us, knowing that this deletion is really highly penetrant for a lot of psychiatric and neurodevelopmental disorders, even though most individuals, we know that they've got this very similar set of genes that are deleted, but there's still a lot of genes in there. So the question is, well, what genes in particular might be driving some of the phenotypes that we're seeing? So that was the question that we had at the time. So we decided to try using an imaging transcriptomics approach to help us prioritize genes within the locus that might drive some of the differences that we were seeing in terms of structure. So what did we need in terms of data? T1 structural MRI data, we had it in 232 patients and 290 controls. We just processed it with FreeSurfer, Desikan-Kiliani Atlas, so like really pretty basic. We then computed sort of the deviance for patients compared to controls across those 64 brain regions from the Desikan-Kiliani Atlas. So that we just knew like what's that map of effect sizes across all these cortical regions. Then we pulled in the Allen Human Brain Atlas data. At the time, some folks who had done some of these studies had kindly made their parcellations of the gene expression data from Allen Human Brain publicly available. So they'd done the work to already map, for example, the transcriptome from all of the Allen Human Brain Atlas samples to the Desikan-Kiliani Atlas, which made it really easy to sort of then start doing these kinds of spatial correlation analyses. So we did a couple things. First, we correlated severity of those phenotypes across regions versus the regional expression of each of the protein coding brain expressed 22q genes to help us prioritize which genes, might be driving these brain differences. And then we also did a sort of a check. We used what's called a partial least square regression, which is a really common analytical technique that people use in this area, which basically identifies a top principal component that maximizes the spatial convergence between the the transcriptomic data and your imaging phenotype. So it's like a principal component analysis, but to maximize the covariance between your across these two different sets of variables. And then we could then also get transcriptome-wide gene loadings onto the top principal component output by this analysis technique. So we could then do a little bit of follow-up bioinformatic analyses to say, okay, like in terms of convergent biology signal, what do we see in terms of like the spatial pattern of differences in 22q patients? What does it seem like it converges on biologically? So beyond just like an individual gene. Okay, I'm going to speed through it and basically say when we ran these analyses, what we saw is out of the 22q genes within that locus, sort of the top gene that showed a pretty high spatial correlation with the pattern of reductions in surface area was this one gene DGCR8. So basically regions that showed greater surface area reductions were regions or greater reductions in surface area were regions where this gene in the adult brain tended to be more highly expressed. And then in terms of cortical thickness, we saw a different 22q gene come up where regions that showed for 22q, their deviance means greater thickness, so higher thickness, are regions where this gene P2RX6 was more highly expressed. When we ran the partially squares regression on all of the Allen Human Brain Atlas data, so not just our 22q genes of interest. For surface area, we saw that the genes that loaded most highly so we looked at the top 5% of genes that loaded most highly on the partially square regression first principal component were involved really broadly in regulating the expression of genes. And DGCR8 from that analysis still came out among the 22q genes as one of the top genes. And same thing with partially square regression for cortical thickness. It was the genes that broadly tended to have a expression profile that sort of mapped onto the spatial patterning of thickening in 22q were implicating transmembrane and ion transmembrane transport. And again, when we checked among the 22q genes out of the genome-wide transcriptome first principal component, we were seeing the same P2RX6 gene come up. I'm going to skip this part, but basically say this was pretty interesting biology when we saw these sets of analyses, and this is just correlational. So we didn't do a follow-up study to then really fully determine, do like an experimental manipulation to really truly get at causality. But what was interesting is that DGCR8 had already been sort of put out there as maybe one of the key genes within the locus that might be driving a lot of the phenotype in 22q patients, because it's actually a gene that is really important for modulating the expression of actually many other genes, including during early fetal development. But then it also highlighted this other gene, P2RX6 for cortical thickness, that really was relatively understudied. So it's creating also some new hypotheses. Okay, so what I'm going to do is I've got some last slides here, and I'm going to, they might already be posted, or if not, they will be posted. So I've just linked here or like put up some resources for you all, like further reading along some of these lines if you're interested. With imaging transcriptomics in general, there's been like really an explosion in sort of this kind of analysis in the last several years. And there's really nice toolbox out there that now make this a lot easier if you've got other parcellations of the brain that you want to work in. There's this really nice toolbox called Abogen that helps you reparcellate the gene expression data from the Allen Human Brain Atlas into whatever parcellation maybe you are interested in working with. And there's also some nice papers that came out sort of talking about sort of methodological considerations if you're working from the raw gene expression data, like, there are going to be some decisions you have to make about how you process that data. So these are really nice resources to give you some hints. And I've also linked, this is like, if you're trying to do those follow-up bioinformatic analyses, we don't have time for it today, but they are easier than you might expect to run. If you've got a list of genes, there are these lovely bioinformatics tools that people put together. You can just plug in your list of genes and see sort of like where there's convergent signal. These are a couple ones that are pretty popular. If you're looking for summary statistics, for example, for psychiatric disorders, the Psychiatric Genomics Consortia website makes a lot of them publicly available. A lot of other studies will list for a different trait. They'll tell you oftentimes that they've made their summary stats available, and they'll tell you where it is. And then I've also linked, or I've also posted a couple papers, like if you just want to sort of dive in a little bit more of like, what are the basic steps of even just a genome-wide association study? This is a really classic old paper about the methods for genome-wide association studies. And on the right is a paper that sort of gives you a really nice overview of just sort of what are the steps that you think about when you're actually creating a polygenic score based on GWAS summary stats. Okay. And with the last 15 minutes, if folks are okay, we can switch over onto the hub. And what I've put up on there are some example scripts. The first one is in R, actually, and it's just like, how do you run a partial least square regression? If you've got that sort of map of effect sizes, I've given you the map of effect sizes that we had from the 22q study for cortical thickness. And then also the map of gene expression data that was parcellated to the same Desikan-Kiliani atlas. You can see some example code for running a partial least squares regression. So we'll start with that. And if we have time, I haven't used the Abogen toolbox in my own research personally, although I have colleagues who have, and we did just get it installed up on the hub. We did run through it. So there's also an example Python notebook for just reparcellating the Allen Human Brain Atlas data to, again, the Desikan-Kiliani Atlas. But you could also, if you have, again, your own parcellation, you can upload that and sort of use some of that code to reparcellate it for your own study of interest. And if you have any questions, please feel free to email me. And maybe I will switch over to the hub and sort of maybe we can talk through some of the steps. But it is hopefully fairly straightforward. You can just pretty much run the code and sort of look at your output to try to understand what's happening at each of the steps. So, okay, let me. Okay, so for the first partial least squares regression analysis, you would just go to the hub and you need to select the R machine because the code that we used was based in R. More recently, there's been like a Python-based package, I think, that has been developed. I haven't used it myself, but if you're interested, I'm pretty sure they exist too.
Just briefly talk about how the field like conceptualizes how 6 samples is representative.
That's a great. Yeah, so 6 individuals, 6 adults, I think maybe all of them are male, or most of them are male. There's some racial, ethnic diversity, but yeah, is the pattern of expression in these 6 individuals, is that representative of the population? Absolutely not. And at the same time, we know that there's much larger variation due to in terms of the transcriptome across the brain, due to developmental stage, for example, and brain region compared to individual. So this is obscuring those individual differences. And you will see like there are metrics that people have computed, like how similar is the expression of a given gene across the 6 samples. It's not, some of the genes vary. There are some genes that show more variation between individuals and some genes that show less. So it is very much, I would say, an imperfect tool. At the same time, it's been pretty powerful. So I think we can still use it knowing that there are limitations to it, like with any of our methods. I think there must be newer initiatives to try to get create sort of similar data sets, like maybe with like single cell RNA-seq, but I don't know, like, and probably hopefully more individuals, but I don't know the details of sort of where all of that is. For now, this is sort of the best data set that I know of, but yeah, definitely with some caveats about.
for.
Oh, interesting.
Maybe represent.
Yeah, yeah. I think that what's been sort of nice is people have done some of these kinds of analyses is that some of the biology you would expect is getting implicated, which I think gives people like some confidence that like, okay, like this isn't total crud, whatever it is, like it makes sense, which helps that sort of like face validity. You do also have the option, for example, to see for yourself if you like take out certain samples for or certain individuals, and you can see for yourself if I'm only using individuals, like half of these individuals, and I randomly sample which ones I use, how much does that change sort of the patterning of gene expression? I feel like somebody must have done that analysis. Actually, maybe the Abidjan paper, they might have even talked about that a little bit. I think they might have done something like that. And it does change things a bit, I think, but again, because so much of what drives gene expression patterns actually is cell type and cell type composition of your bulk tissue. That is like the biggest signal. And what the cell type composition is that you have in different regions of the cortex or cortical regions compared to subcortical regions, you're going to have pretty different cell compositions. I think that's still going to be the largest signal. Yeah. Yeah. But something you can empirically check too, which is really cool. Yeah. So if anybody wants to, I think you could play around with the Abidjan toolbox and see it for yourself. Okay. Oh, I'm sorry. I realize I'm not sharing my screen anymore. Let me reshare. Did everyone get to the R script? Okay. Okay. Let me. Sorry, let me fix my Zoom. Do I need to? Oh, there we go. Okay, there we go. Oh no, I'm not gonna be able to see it on my screen. I think it's this one. Okay, so if you've got an R machine, you can go to curriculum and Foresight Genetics, and then this example R script, we can sort of walk through together. Sorry, with my current settings, I can't actually see it on my own screen, so I'm gonna turning around. I don't know if there's a way to duplicate screens. Okay, I'll just turn around. Okay, so you need to load some key packages. Nope. Oh, thank you. Oh, did I not do RStudio? I'm sorry, hang on, that was my bad. So do RStudio between your options. I'm gonna pull this back over for myself. I'm sorry, y'all. Also, feel free if you want to just start running through the script. That's okay too. And you can start looking at the output, but otherwise I will, well, maybe I'll just talk through it. I think if you all hopefully loaded it as an RStudio environment, you've got like normally your R script, and then you've got your console, and you can see your variables in the upper right. Did people do that? Awesome. Okay. So then you need to load these packages and set your working directory in a variable for the folder where the data is. That's through lines 15 and 16. Hopefully everyone got through that okay. Then if we go down to line 23 and you execute that, that's gonna load in a matrix that contains the gene expression data from the Allen Human Brain Atlas that was made publicly available from a 2015 paper by French and House, where the data had been, again, mapped to the Desiccant-Tiliani Atlas already. And in our analyses, we were focusing on genes that were expressed in brain above a certain level and that were protein coding. So it was subset a little bit compared to sort of the original data set that was made available. So if you load that in line 23, starting in line 26 through 28, those are just some commands to help you start looking at what the data is. Like, what are your rows? What are the columns? How many genes do you see in there? How many brain regions do you see? Yeah.
I'll just be maybe, I'm not saying.
And if you hit the, if you run that command, it's not loading it in.
I'm also very.
So notice that the R code and the data are in two places. One is in curriculum, the other is in shared. So that's why the code is pointing to the data folder. And the data should be in the code. So is anyone able to run the code in RStudio? It's not long. I think maybe the difference between those one and is where RStudio was launched. And the suggestion is to navigate in Jupyter Lab to the home directory and then launch RStudio from there. I think in some places you might not have to, in the hub, you might not have permissions to place. Okay.
Okay. Sorry about that little hiccup. Hopefully most people are able to load it. From there, does somebody want to holler out how many genes it looks like there are in this matrix? Yeah, a little over 10,000, thanks. And how many brain regions do we have? Yeah, awesome. So that's because here, like I was mentioning with the Allen Human Brain Atlas, not all of the 6 individuals had right hemisphere regions. So for our analysis, we just focused on using the median expression values that were derived across all 6 individuals on left hemisphere. So we just ran basically left hemisphere based spatial correlation analyses. And then if you run line 32, that's going to load in the map of the effect sizes for our 22q patients compared to our controls in that same set of left hemisphere Desiccancilium atlas regions. You can see the effect sizes in one of the supplementary tables from the paper. So we're just loading in that map of effect size differences for those same 34 regions. You run line 35, that's just giving you a little bit of information about what's contained in that object that you've just loaded in. And then from there, are folks able to get that one in okay? And then from there, if you run line 38, it's basically combining those 2 sets of data. And we've already checked it, but the ordering of the regions has to be the same in the 2 sets of variables, right? Because you're running a spatial correlation. So you would want to make sure that the ordering is actually the same. And then you put them in one data frame together, where the first part of the new data frame is just that effect size difference across regions variable. And then the second part of it is a variable that actually contains an embedded matrix of the gene expression for those 10,000 or so genes by 34 brain regions. You can run like an STR on that object if you'd like to, look at what it looks like. And then that's really all you need before you actually run the partial least square regression. So in line 44, we're using the PLS package for this, which has been around for quite a while. There's nice documentation on it, really nice tutorials from their package about how to do partial least square regressions. So if you run line 44, it's going to do that partial least square regression for you. And we're running it with leave one out cross validation. So it's basically checking. We haven't specified how many components we think the model should have. So it's basically going to compute sort of error metrics for the model each time leaving out one of the brain regions. And if you run line 47, you can sort of see some of those metrics about your model fit. And then if you run line 50 through 52. Can also run 55 through 57. That's just going to generate some PDF plots that you can then download to your local computer and look at to basically see. It's just plotting some of the like the root mean square error of the different models, depending on how many components a given model has. Or the amount of variance explained R squared by each of these models. And then what you can basically do is say, okay, I want to pick the model that has the lowest error, which in our case would have had 3 components. The majority of the time in these kinds of imaging transcriptomic analyses, they're mostly just looking at the first principal component that's explaining the most variance anyways, but if you wanted to look at additional components, you could do that as well and characterize them too. And then when you get to line 64, you're just rerunning partially square regression now, specifying that you want the model that had 3 components. Lines 67 through 69 are just computing the observed root mean square error and variance from this final model. You would cite some of those metrics maybe in a paper. And then I think what gets really interesting is when you get to line 72 and run through line 78, what you're doing is you're saying, okay, for that first principal component from the partial least square regression, the first component that explained the most covariance between my spatial pattern of effect sizes in my 22q cohort here compared to controls, and the transcriptome, you're getting how much does each of those 10,000 genes in the Allen Human Brain Atlas that we kept in our analysis, what's their loading onto that first component? And then you can save that out for downstream analyses. So like I said here, you would then take that list of loadings, you would sort it so you're looking at sort of the genes that load the most on that first component. A common strategy would be to take maybe the top 5% of those genes and then hop over. There's some R packages to do some of these functional enrichment analyses. There's also really nice like point and click like web GUIs to do some of these bioinformatic analyses. To get to run, for example, a gene ontology enrichment analysis, where basically lots of people over many years have compiled lists of genes that are annotated to different cellular, molecular, biological processes in the literature. And so you can then mine, mine those associations from the literature through these gene ontology analyses. These tools, if you put in a list of genes, will sort of spit out whether or not your list of genes is enriched for specific molecular processes, biological processes, cell types, things like that. I've given you a link for a tool, for example, that we used. You could also, for example, do the same thing, take those top 5% of genes. There's a tool from the Doherty lab, this PSI tool, where basically they have lists of genes that show cell type specific expression. So you can basically say my list of genes that loaded really strongly in my top principal component output from the partially square regression, are they enriched for genes that have specific expression in certain cell types? So really easy point and click kind of bioinformatic tools that you can use once you have a list of genes. So this is pretty simple analysis, really. For our purposes, we also then intersected those gene lists with our 22q gene list. That's what's down here. So we could, then look at how our 22q genes, like I was showing you, weighted, like where they were weighted in terms of that top principal component. And that's kind of the bulk of the main analysis there. So hopefully you all got through a good chunk of that code and feel like you could run this if you wanted to in some of your data. I'll just highlight down here that what's here is another version of it. If you reprocess the Allen Human Brain Atlas to a different parcellation using, for example, the Abidjan tool, there is a script here, this one, which I don't think I can load because I'm in RStudio, but if you switch to a different instance of your Jupyter notebook, like just like the normal CPU instance, I think, you can then, Abidjan is a Python-based tool. There's just example code using the Abidjan tool to reparcellate the Allen Human Brain Atlas data. And we've annotated that. So maybe if you're interested in this, like next week during the hackathon week, you could try that out. You could try, go into the Abidjan documentation, and if you've got your own parcellation that you're interested in, try parcellating to your own parcellation. And then you can load back in that data. Again, check your labels, make sure they're like all ordered the same in your effect size map and the expression data, but then you can run a partially square regression on your own data. Okay, I'm sorry, I'm realizing I'm over time. I'm sorry about that. Thanks everyone so much for your attention. And again, feel free to email me if you have questions, happy to chat.